In type 1 diabetes mellitus, the immune system destroys pancreatic beta cells. With no insulin present, glucose entry into skeletal muscle is markedly reduced. Glucose levels in the extracellular fluid, the ECF, rise, and we detect this as hyperglycemia on blood testing. Also, in the absence of insulin, unopposed release of glucagon further aggravates the rise in ECF glucose by the mechanisms we have previously discussed. As glucose is an osmolyte, and as it cannot enter cells in this situation, hyperglycemia is accompanied by a rise in the osmolarity of the extracellular fluid relative to the intracellular fluid. This rise in ECF osmolarity has a number of important consequences. The rise in ECF osmolarity will cause a shift of fluid from the intracellular to the extracellular fluid compartment, with consequent cellular dehydration. In addition, osmoreceptors in the hypothalamus sense the rise in ECF osmolarity. A rise in ECF osmolarity deforms these cells by inducing movement of water from their cytoplasm to the extracellular fluid. In response to this, osmoreceptors then signal to neurons in various nuclei of the hypothalamus, and these neurons respond by increasing the release of antidiuretic hormone from the posterior pituitary into the systemic circulation. The ADH released into the systemic circulation acts on the kidney. ADH promotes the insertion of aquaporins into the collecting duct epithelium. In this way, ADH promotes absorption of water, free of sodium, from the lumen of the collecting ducts back into the renal parenchyma, along the osmotic gradient between these two sites. This water is absorbed from the renal parenchyma into the capillaries and is returned to the general circulation by the renal venous system. Osmoreceptors also project to thirst centers in the brain. Thirst mechanisms will be activated in the presence of raised ECF osmolarity, increasing free water intake. All of these effects, the shift in water from the intracellular to the extracellular fluid compartment, ADH activation, and increased oral fluid intake, will tend to normalize plasma osmolarity, but may also result in dilution of ECF sodium, with significant hyponatremia developing on blood test analysis.